In this video, we will discuss about this NK cell invasion with CD-44 neutralization. So far, we have discussed the results that are presented in this paper and come to a hypothesis that the CD-44 expression on the NK cells interact with the collagen, fibroblast that are more commonly expressed in the tumor microenvironment of the pancreatic cancer cells, and that prevent the NK cells to invade and attack the tumor cells. So in order to test that hypothesis, what they did is they neutralized CD44 and check the NK cells invasions. So they did their studies in 2D model and also in the 3D model. We will discuss both the results in this video. So this is their experimental plan of the 2D models, where what they did is they take the NK cells and treat and in one conditions they use this anti-CD44 to neutralize the NK cells, and in other conditions they don't use anti-CD44, and they coat this transval migrations with the material and collagen mixture, and in the outer layer they use this cytokine, that is CXCL9 of 100 nanogram per ml, and they did their study on 4 different human donor cell lines. And what they are able to find out is that when they use neutralizing CD44 antibodies, they can see more NK cells invasions, that are able to pass through this membrane, compared to the case where there is no and neutralizing and anti CD44, there is no neutralizing CD44 antibodies are present, and this increase is statistically significant for 3 cases out of the 4 cases that they have tested. For the fourth case, it is there looks like an increase, but this is not statistically significant, so they don't consider it as a increase. And once they get confident that their hypothesis seems to be correct, they wanted to check this behavior in a 3D model. So what they did is they develop a spheroid of Pancreatic cells and then culture them with the donor NK cells and the media that contains either anti-CD44 antibodies or no neutralizing CD44 antibodies. And the surface that they use in this 3D model is a mixture of 80% collagen and 20% material. And in order to stain this spheroid, they use Penn's cytokeratin, and they are able to see that when they don't use neutralizing CD44 antibodies, they can see different spheroids or smaller spheroids, whereas when they use anti-CD44 antibodies, they do not see too much fragmentations of the spheroid. Or we can say that the objective of this Penn cytokeratin is only to mark the spheroids that are presented within the samples. So here they have marked the spheroids, different spheroids, and in order to stain the NK cells they use CD56 markers. And what they are able to see is that in the case where they don't use these neutralizing CD44 antibodies, they do not see any CD56 infiltrations within the spheroids or Pancreatic spheroids, but where they use these neutralizing CD44 antibodies, they can see the spheroids that are marked by white arrow, and CD56 is conjugated with green fluorescence. It can be seen here, and this is the zoom in image where we can see that the spheroid, the CD56 signals more clearly, or NK cell signals more clearly within the spheroids, but this kind of signals are not visible in the case where neutralizing CD44 antibodies are not used. And the quantification results are shown in this panel IJK about the 3 donors, and here we can see that when they use neutralizing CD44 antibodies, there is a increase in the NK cells within the spheroid for these 3 donor cases, and so far I am not sure whether they did the study for 4th donor or not, but the result I have presented here for the 3 donor studies, and in all the 3 cases we can see that the invasions are more when they use the neutralizing CD-44 antibodies. So this further substantiates the previous hypothesis that CD-44 might be interacting with the collagen fibronectin that are overly expressed or present in the tumor microenvironment of the pancreatic cancer cells, and thereby prevent the invasions of the NK cells to interact with the tumor cells and trigger immune response. I hope you understood how they validated their hypothesis using 2D model and 3D model. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.